All right, are there any questions from last time? So everybody up to date with whatever we have covered, right? So, I mean, you're ready to take a test, right? I mean, that's the level of proficiency that you should have. I'm covering quite a bit of material, but, I want you to, really be up to date. Think like you're going to take a test in the next class, all right? That's the only way to absorb so much of material, especially if you have had almost zero background in biology. Right? It's not difficult, but you have to put in a lot of work, right? So any questions about proteins and so we discussed proteins, we talked about energy considerations and biochemical reactions. Any questions on those? Otherwise, we'll move on to the next chapter, which is about DNA, right? So DNA is the abbreviation for deoxyribonucleic acid, right? Because the sugar in DNA is deoxyribose, right? I already covered that. We'll talk about that again today. All right, and DNA is the molecule that is used in cells to store genetic information. Now the genetic information must be passed on from a cell to its daughter cells at cell division, right? So every time a cell divides, the entire set of DNA has to be copied, right? And then properly apportioned between the two daughter cells, right? And this information is passed on from generation to generation of organisms through the reproductive cells, right? You get your DNA from both your parents, right? Who in turn got it from their parents and so on. Okay, so it's being handed down through generations, right? And in a very faithful way. So, so what are genes, right? Genes are the information containing elements that determine the characteristics of a species as a whole and of the individuals within it. And the natural questions that come up are, I mean, today in 2016 we talk about genes, and we say it's in our genes, right? But we need to know, right, what is the composition of the genes, right? And so so there are several questions that come up. What is the chemical composition of the genes, right? And what is the mechanism by which the genes are capable of storing information which can be replicated in an almost unlimited fashion, okay? Because it's across thousands of years, right? That information is getting handed down, right? Now, by the early 20th century, right, biologists knew that genes were carried on chromosomes, right? Because they had seen chromosomes. When a cell is dividing, they can see the DNA contents, so they knew what chromosomes were, and they knew that genes were carried on chromosomes. Now, biochemical analysis of chromosomes revealed that chromosomes are made up of DNA and protein, right? And we have already talked about proteins. They are basically amino acids linked together. DNA, these are strands that are made up of those nucleotides that are linked together by what kind of bond? What kind? I didn't get you. No, peptide is in proteins. Okay, what about the DNA molecules? I mean, the nucleotides are linked together by what kind of bonds? What bond? No. Anybody? What? Yeah, go ahead. Not in the nucleotides, no. A nucleotide is made up of what? That's the base, okay? What else? So it's made up of the nitrogenous base, then the sugar, right? And then the phosphate, right? Exactly, yeah. So phosphodiester bond. See, you have to keep on studying. It's okay, even if you don't get it in one class or two lectures, but let's say 2 weeks from now or 1 week from now, these things should be at your fingertips because these are the things that are going to show up on the exam, right? And you cannot memorize it the night before the exam, right? So I would urge you to really go over each lecture as it is delivered, right? And come up to speed. A little bit, of course, you'll lose out, but you have to make a really serious effort. Otherwise, it's going to be a disaster. Right? So chromosomes are made up of DNA and protein. So initially, people thought, right, that the carrier of genetic information would have to be the protein. Why? Because DNA looks like a pretty simple molecule because in each location, right, there are only 4 possibilities, right? A, G, C, T, right? Whereas if you look at a protein, it's made up of amino acids, and at each location there are how many possibilities? 20 possibilities. Okay, so that looks like a much more complicated molecule, that could store the genetic information, right? So this notion was put to rest by an experiment that was carried out in 1944, right, which I'm going to discuss next. And this experiment conclusively proved that DNA is the carrier of genetic information, right? So in this experiment, scientists took 2 closely related strains of the bacterium Streptococcus pneumonia, right? So this is a bacterium, that cause pneumonia in animals, right? And there are two different strains, okay? One of the strains of this bacterium is called the R strain, and R comes from the word rough because it appears rough under the microscope, right? It is not pathogenic and doesn't cause pneumonia when injected into animals, right? So this is like the good strain, right? If there is anything like a good strain of the bacteria, right? The S strain or the smooth strain, right? And again, the S comes from the word smooth, right? Because it looks smooth under the microscope. This is pathogenic, right? And it causes pneumonia when injected into animals, resulting in death, right? So the experiment that people did was the following, right? You basically go and take some S strain bacteria, right? You kill them by heat so that bacteria cannot multiply anymore, right? Or cause pneumonia, right? Then you grow R strain bacteria, the seemingly harmless bacteria, right? In the presence of that heat killed S strain bacterium, right? And then see what happens. So live R strain cells were grown in the presence of heat killed S strain cells, okay? So the bad guys had been killed by heat, right? The S strain cells. Now the animals that were injected with these cultured R strain cells died, leading to the conclusion that somehow the R strain cells had been transformed to the S strain cells. Okay? Because the good guys had now become bad guys by growing in the company of the bad guys. Okay? So the first conclusion is that the molecules that carry heritable information are present in the heat killed S strain cells. Okay? Although they are dead, there is some information that can be passed on. Right? So that's the first conclusion. Right? Now to find out which traits are the ones that really got passed on, the cell-free extract from these transformed R strain cells was fractionated into classes of molecules, right? And the resulting RNA, proteins, lipids, and carbohydrates, all of them were found to be of the R strain type. Okay, nothing had changed. They were still of the original type. The only thing that had changed was the DNA. The DNA was found to have been transformed to the S strain type, right? So now the DNA, now you have the DNA of the bad guys, basically, right? So the immediate conclusion is that the DNA is the molecule that carries the heritable information, right? It's not the protein, yeah.
So when you say like you kill the S strain bacteria, right? So does that mean that it's chemically inactive or?
No, that means the cell is dead, okay? Only the components are there, okay? It's not live, like it's not capable of reproducing or performing any of the cellular functions. But the molecules are still there. Yeah.
So that's how it got transmitted.
Yeah, I mean there is a mechanism. It's called transformation, by which the bacteria can take up like DNA from its surroundings. All right. So that's how it got transmitted. Yeah. Just one moment. So the conclusion is that DNA carries the heritable information. So any questions so far? Yeah.
This is an odd question I have. So, how did they? I mean, I guess it's completely irrelevant, honestly, but I was wondering how they would know if the RNA and the proteins, all the other things were from the R strain bacteria, if the DNA from the S strain bacteria was obviously producing those things in the new bacteria.
Yeah. How would they know that?
Yeah, if
They could probably label. I don't know exactly how they did it, right? But today, if you want to track DNA, right, you can put some radioactive isotope or something like that and track it. I don't know how they did it in 1944. I mean, you'd have to probably look at the history, and this experiment has a name, right? So you look up 1944 maybe on the internet you'll find it, right? I don't know the details of how they exactly figured that out.
yeah, that's probably what they did. They used radioisotopes or something like that.
Yeah, probably. But you have to remember one more thing, that at that time, well, I guess they probably, no, they didn't know the detailed structure of DNA, but I think, they didn't know the actual, what should I say, three-dimensional structure of DNA, how it's arranged, but they probably knew the composition. So yeah, they could have probably produced some radioactive isotope, incorporated some radioactive phosphorus into the DNA that they were producing for the S strain. That's my guess, but I don't know for sure. Okay, because remember this is 1944. Okay, so it's like what, 70 whatever years earlier. I have not.
But that makes perfect sense. I mean, the other things were obviously produced in that cell and would
Yeah,
not have been radioactive.
yeah.
DNA
Okay, any other questions? Now, the structure of DNA was discovered by James Watson and Francis Crick in 1953, right? So again, I mean, and they got a Nobel Prize for this, right? And the difficulty, I mean, the Nobel laureate Linus Pauling also worked on it, right? But these guys got it before him, okay? Because they built a model using sticks and chewing gum, and so you have these four bases, right? A, G, C, and T, right? How are these things arranged, right, to form the DNA molecule? So the first thing is you have a nucleotide. That's the DNA base, right? Then you have the sugar, right, which is in this case is what is the sugar for DNA? Deoxyribose, and then you have the phosphate, right? Now, the sugar, so the phosphate is attached to the 5' end of the sugar, of the deoxyribose, and at the 3' end, there's a possibility to attach something else, right? So now you have a nucleotide that is made up of the DNA base, the sugar deoxyribose, and the phosphate, right? So 5' is at this end, and 3' is at this end, okay? So these kind of units, you can link them together, right, end to end, right? And you get a single DNA strand over here, right? And this is the sugar phosphate backbone at the top, right? And that portion is the same all throughout the DNA strand, right? So that's step number one. Then you'll have two strands, right? That are where the DNA bases are going to be complementary to each other. G on one strand is always going to pair with C on the other through 3 hydrogen bonds, right? You can see that here. A is going to, on one strand, will pair with T on the other through 2 hydrogen bonds, right? And so now you have a ladder, right? The rungs of the ladder are made up of those DNA base pairs, right? And then on either side, the sides are made up of the sugar phosphate backbone, right? And then this thing twists around itself, right, to produce something that looks like a spiraling staircase, right? And this is the famous double helix that you see all over the place, right? You don't have to take this course, you look at Time magazine, 50 years of the double helix and all that. There are issues that have come out in that, right? Now here I just want to, this is something we'll mention later, but I want to point out that keep in mind that A pairs with T through 2 hydrogen bonds. G pairs with C through 3 hydrogen bonds. So if you needed to take the DNA strand apart, okay, which would be the easier location to break it apart? Would it be an AT bond or would it be a GC bond? AT bond, because it has fewer hydrogen bonds. Okay, so just keep that in mind. All right. We will talk about this when the strand comes apart. There are what are called replication origins, which will be the locations where A and T are linked together. Okay, any questions on this? So this was like a grand challenge, and they figured this structure out in 1953, But once they got the structure, then the replication mechanism also became quite clear. Because all you need to do, because one strand has information that is complementary to the other one, okay? So for replication to happen, you can take the two strands apart, right? Each strand can serve as a template for the other strand, right? And that's how DNA can be replicated, right? And we'll discuss that in details. So a DNA molecule consists of 2 complementary chains of nucleotides, and 4 different nucleotide bases make up DNA, right? Adenine, guanine, cytosine, and thymine, right? A, G, C, and T, right? A always pairs with T through 2 hydrogen bonds, while G pairs with C through 3 hydrogen bonds. Now, the pairing of A with T and G with C ensures that each pair is of similar width, okay? So that you have a nice ladder, okay, where the width is the same all throughout, right? And thus holding the sugar phosphate backbones an equal distance apart along the DNA molecule. And in addition, as I said, the 2 sugar phosphate backbones, they wind around each other to form the famous double helix of DNA. All right, any questions? Now, DNA encodes information in the order of sequence of the nucleotides along each strand. Each base A, G, C, or T can be considered as a letter in a 4-letter alphabet that is used to spell out biological messages in the chemical structure of the DNA. Organisms differ from one another because their respective DNA molecules have got different nucleotide sequences. You may have heard of the Human Genome Project, all right? So if you take a human cell, you look inside the nucleus, there are 3 billion of those letters, all right? So the Human Genome Project basically went ahead and sequenced that entire thing, all right? Right? Let's say for a human cell. And it took like, I think, about $4 billion and 10 or 13 years to do it. Today you can do it in 24 hours, and you can do it for $1,000, right? So, yeah. It's still expensive, but, for $1,000 you can get your DNA sequenced. So organisms differ from one another because their DNA, those sequences are different, right? But in those 3 billion letters, That is the entire information to make a complete human being. So the linear sequence of nucleotides in a gene, right, that means all that A, G, C, T, basically spells out the linear sequence of amino acids in a protein, right, because the DNA codes for proteins. And the exact correspondence between the 4-letter nucleotide alphabet of DNA and the 20-letter amino acid alphabet of protein, which is called the genetic code, will be discussed in the next chapter, right? So there is you can get a map, from DNA to protein, right? And again, I mean, people that discovered those, they also got Nobel Prizes because these are, profound things, okay? They have changed the landscape of biology, right? So in this chapter, we are going to focus on the mechanisms by which DNA is replicated, right? Possible errors that might take place during DNA replication, because, if you make mistakes, that can be catastrophic, right? And the mechanisms that are used to correct them. Now, most genes are short stretches of DNA encoding a single protein. So as I said, in the seminar that I gave, that roughly speaking, a gene is a stretch of DNA that codes for a protein, right? However, not all of the DNA in a gene is used to code for the protein, right, that it specifies. There are also regulatory regions which will specify when and in what amounts the protein that is encoded by a gene is going to be made, okay? Because you don't need the same proteins all the time. The cell doesn't need the same. So the genes coding for a particular protein should be turned on only in response to the needs of the cell. Now, the complete set of information in an organism's DNA is called the genome. So if you're talking about the human genome, it is the 3 billion letters, okay, that are present in the DNA of a human being, right? And it's exactly the same DNA, whether you take the skin cell, the liver cell, the heart cell, whatever, right? It has exactly the same DNA. At each cell division, the cell must copy its genome in order to pass it to both the daughter cells, right? Makes sense, right? And each strand of DNA contains a sequence of nucleotides which is exactly complementary to the nucleotide sequence of its partner strand, right? Therefore, each strand can act as a template or a mold for the synthesis of a new complementary strand. And that's shown on the next slide, right? So you have two strands, S and S prime, right? If you know the sequence here, you automatically know the sequence here because A on this will pair with T on this, C on this will pair with G on this, and so on, okay? So the strand S can serve as a template for synthesizing a new strand S prime, right? You can use this to get a new strand S prime and vice versa, right? Any questions? So how is DNA replicated, right? So the DNA replication scheme goes as follows. First, the two strands separate out, right? Then each strand can be used as a template for synthesizing its complementary strand, right? And then the newly synthesized DNA strand and the template used to create it, they'll pair together to form a new double helix, right? So every time you have replication, you've basically multiplied the amount of DNA by 2. Okay. Now DNA replication is semi-conservative, right? And again, this was established through experiments, right? I'm teaching all this stuff to you as though, that's the only way. There are other ways, right? But people have done experiments, right? Again, with the kind of labeling and all that stuff to see, is it semi-conservative or is it that the original strands pair back together, right? And the new strand, the new double helix is completely new, right? People have done experiments to figure out what the actual situation is, right? And from those experiments, they have learned that replication is semi-conservative. In each round of replication, one of the strands that you get in each double helix is conserved from the previous round of replication. Now, replication begins at replication origins, right? Where the two DNA strands separate. And this separation of the two DNA strands is usually carried out by initiator proteins that bind to the DNA and pull the two strands apart. Okay. And since as I said before, since DNA that is rich in 80 base pairs has got fewer hydrogen bonds, it is usually the site of replication origins. So if you look at a bacterial genome, right, if you look at the genome of a bacterium, right, that is usually small, maybe 1,000 genes or something like that, right, or maybe even less. So usually it will have only one replication origin, right. But in the case of the human genome, right, you have 3 billion letters, right. There has to be a lot of parallel processing. So in a human genome, there could be as many as 10,000 replication origins, right. So simultaneously the replication can start, right. and the entire genome can be replicated quite fast. Now the core of the DNA replication machine is formed by an enzyme. All right. Again, what are enzymes? Enzymes are proteins which are? Okay, good. So the core of the DNA replication machine is formed by an enzyme which is called DNA polymerase. Okay, and we introduced this terminology before. Polymerase means it produces a polymer. In this case, it will produce the DNA, polymer of DNA. Right. This enzyme will synthesize this enzyme synthesizes the new DNA using one of the old strands as a template. Okay. So this enzyme catalyzes the addition of nucleotides to the 3' end of a growing DNA strand. So this is the 5' end of the DNA strand, this is the 3' end, right? So DNA polymerase will cause growth in this direction, right? The next nucleotide will be added here and then the next one here and so on, okay? So it will grow only from the 5' to the 3' direction. It does not grow in this direction. You cannot grow like this, right? So this enzyme catalyzes the addition of nucleotides to the 3' end of a growing DNA strand by the formation of a phosphodiester bond, right, between this end and the 5' phosphate group of the incoming nucleotide. And to make the reaction possible, energetically favorable, the nucleotides will enter the reaction as energy-rich nucleoside triphosphates, right? Because we know that this has got a high energy bond here and another high energy bond here, right? Because remember, producing DNA from nucleotides, this is an energetically unfavorable reaction. Okay, that's a reaction that would not normally happen. That's order generating, right? So you have to couple it with something that produces heat, right? Or some other reaction that is energetically favorable, right? So that's why it will enter. So here you can have a DNA base. It could either be adenine, guanine, cytosine, or thymine. It could be have a 6-member ring or a 9-member ring linked to the 5-carbon sugar deoxyribose. And then instead of it being a monophosphate, it's going to be a triphosphate here. So that these bonds can be broken down to make the reaction possible. Right? So the hydrolysis of one phosphoanhydride bond in the nucleoside triphosphate provides the energy for the condensation reaction that links the nucleotide monomer, right? That means with only one phosphate, right, to the chain and releases pyrophosphate. So first, this bond here is broken, right? So then you have the monophosphate here that gets attached, right? The phosphodiester bond is formed, right, here, and then you have pyrophosphate, right? Two inorganic phosphate ions linked together. Again, this has high energy bond here, right? Then this is also hydrolyzed, right, to produce two inorganic phosphate ions. So in the process, I mean, you've done all these reactions, you've made the process basically irreversible, right? Because you don't want the chain to fall apart, okay, as soon as it is formed, right? So the energetics are such that you have made an unfavorable reaction go through, right, and made it irreversible, right? That's how the chain is going to grow. Any questions? Now there are some complexities associated with DNA replication, right? And we are going to take a look at them one by one, right? Now the first thing is that, so you have this original double-stranded DNA, right? This is 5' to 3' direction, 5' to 3', right? On one strand, 5' to 3' on the other, right? Now you have a replication origin, let's say here at the center, right? So initially the strand comes up, the two strands come apart over there, right? So at the replication origin, and then the rest of it is also coming apart, right? So starting from the replication origin, the strands are coming apart in both directions, right? So now see what happens, right? When you're trying to use these guys as templates to synthesize the DNA, see the problem that we run into, right? So you have the 5' to 3' strand, right? Then you have the 5' to 3' strand. This is the replication origin at the center, right? And then you have the two strands have come apart, and then this thing is propagating in both directions, right? So now if you start your DNA synthesis at the replication origin, right? So let's say you're synthesizing the complement of the top strand here, right? So this strand is being synthesized from the replication origin as you're going to the left from in the 5' to 3' direction, right? The top strand is 5' to 3', this is going 5' to 3'. So as the thing opens up, right, from the replication origin, as this is called the replication fork, as this opens up, this strand can be synthesized without any problem because you are going in the 5' to 3' direction, right? And DNA polymerase can synthesize only in that direction, right? Same thing is true for this bottom strand here. If you start from the replication origin, Again, going to the right, you're going from 5 prime to 3 prime, you're synthesizing, so you can synthesize it continuously, right? On the other hand, if you look at these guys here, right? See, let's say you started here, then you synthesize this, all right? Now, in the meantime, this stuff here has opened up, right? But you cannot go and synthesize in the 3 prime to 5 prime direction. So you have to somehow jump from here, again synthesize from 5 prime to 3 prime. So you see there's a problem. So this is called the leading strand template. This is called the leading strand, right? Where continuous DNA synthesis can take place, right? On the other strand, this is called the lagging strand because you have to synthesize it discontinuously, right? Because DNA synthesis can only take place in the 5 prime to 3 prime direction, right? So here and also here, you have to synthesize it discontinuously, okay? And these have a name. These fragments that have to be synthesized discontinuously, they are called Okazaki fragments, named after the scientist who discovered them. Right? But there is more work coming because now you have a continuous strand here, you have a discontinuous strand over there. Right? So somehow you have to make it continuous, and there are additional steps that are involved. Okay, before that, any questions? So this is called the replication fork, and they say it's asymmetrical because here you have to do it discontinuously, whereas here it is continuous. Is possible. Do you see that? Yeah, go ahead.
What do you mean by continuous and discontinuous strand?
No, the thing is, if you start the DNA replication here, for example, right, and as this thing is opening up, right, you are going to synthesize from right to left, right? I mean, so there is no interruption. So as the strand is opening up, as the fork is growing, if you start it from the middle, you can just keep on synthesizing, right? So that is the continuous strand. You don't have to interrupt things. But on this one, look, see on the other hand, let's say the DNA replication started here, for example. So you're going in the 5 prime to 3 prime direction, you synthesize this portion, right? But this thing is opening up, right? Now from here you cannot go and synthesize in the opposite direction because the synthesis has to be in the 5 prime to 3 prime direction. So that means you have to jump from here, right? The DNA polymerase has to jump from here to this starting point and then synthesize in this direction from right to left. Okay, so that's why this is discontinuous. You cannot just start and keep on synthesizing, all right? I mean, if you started from here, let's say you synthesize up to here. But then again, all of this stuff opened up, right? So you cannot continue, you cannot go in the reverse direction. So you have to jump to that position and then synthesize, okay? Think about this some more, like, I mean, this is something I ask on the exam too, okay, to explain how Okazaki fragments are formed, right? Okay, any other questions? So this stuff is explained in the text here, what I explained in the figure. So DNA polymerase can synthesize DNA only in the 5 prime to 3 prime direction, right? And this causes a problem since the replication fork is asymmetrical, right? I showed that to you. You cannot do it in both directions. Now the DNA strand whose 3 prime end has to grow as the fork moves outward from the replication origin, right, is synthesized continuously, and this strand is called the leading strand, right. On the other hand, the DNA strand whose 5 prime end must grow as the fork moves is synthesized discontinuously, and this strand is called the lagging strand, right? And that's shown on that picture that I used a few minutes ago. Okay. Any questions on this? Yeah, it is. It is creating complementary. It is basically looking at this template, and then it is creating the complementary DNA over there. Okay, but yeah.
So I mean, so you have on the right you have 5 prime first, and then towards the extreme right you have 3 prime. So
No, this 3 prime is for that red strand. Okay, that 5 prime over here, this 3 prime over here is for that red strand over there. Okay.
so why can't it proceed? Because the 3 prime can proceed to the left towards the 5 prime, but the 5 prime can't go close to 5.
Did you get it? Yeah, it cannot do it. But again, think about it, if you don't get the complete thing right now, try to do it, you'll see it doesn't work, correct?
Yeah, because don't forget that there's a 5 prime and a 3 prime on every base pair. That's why you have to really
Yeah. And actually, it's a little bit more complicated than that, than even just doing the Okazaki fragments, and I will get to that additional level of complication. See, DNA polymerase again is a self-correcting enzyme, okay? Because, remember DNA has to be replicated with great fidelity, right? So DNA, it has to check its work, right? So it has a proofreading activity or an error correction activity, right? So it's not just, I know many of you are in communications, but it's not just in communications that you're doing error correcting codes, okay? Biology has figured that out a long time back, right? So before the enzyme adds a nucleotide to a growing DNA chain, it'll check whether the previous nucleotide added has been correctly base paired to the template strand, right? Because if the previous one is messed up, you shouldn't be building more, right? You go and check your earlier work, right? So if the previous nucleotide added has been correctly base paired to the template strand, the polymerase will add the next nucleotide, right? Next complementary nucleotide. If not, the polymerase will remove the mispaired nucleotide and it will try again, right? So it goes back and checks its work, right? So thus DNA polymerase has got a 5' to 3' polymerization activity because that's the direction in which it grows, and it has a 3' to 5' nuclease activity. Remember we said enzymes that degrade nucleic acids are called nucleases, alright? So in the 3' to 5' direction it basically takes out nucleotides or it degrades nucleic acids. So because of this there is a problem, because if every time you go and check your previous work before you do something more, right? You have to have some previous work, right? You cannot start from scratch. When you're initially starting, you don't have any previous work to go and check, right? So there's a problem here. Okay, any questions? So because of this, right, proofreading activity, DNA polymerase cannot start a completely new DNA strand from scratch. It cannot, because it's going and trying to check its previous work. There's no previous work to check, all right? So what is it going to do? So that's why to get the DNA replication started, you need a different enzyme to start a new DNA strand. And this enzyme does not synthesize DNA, but it synthesizes an RNA strand, right? About 10 nucleotides long, using the DNA strand as a template. Remember this base pairing. In DNA you have A, G, C, T, all right? What are the bases that you have in RNA? C and U, uracil, yeah. And U is similar to T in terms of the pairing properties, all right? So this enzyme this particular enzyme, the one that gets the DNA strand started, it synthesizes RNA, because looking at the DNA, you can get the complementary RNA and put that together. And that doesn't have to be that accurate, because you're going to get rid of that initial thing later on, right? For DNA, it has to be very, very accurate. So that enzyme doesn't have any proofreading activity, so it'll get the DNA strand started. Like maybe in the beginning there will be like 10 nucleotides of RNA, and then the rest of the DNA will be synthesized. So this RNA strand is called a primer, and the enzyme that synthesizes it is called a primase. Right? It produces an RNA primer. Okay. So again, let's go back to our earlier picture. You have the replication fork, all right? The top strand is 5' to 3', the red strand from left to right, bottom strand 5' to 3' from right to left, right? And the replication fork is moving away from the replication origin in each direction. So here, if you look at the leading strand, right? Here, once you've primed it once, right? It's continuously synthesized. You don't need to prime it anymore. So there's a primer that will lay out those 10 RNA nucleotides, and then the rest of it will proceed to completion as the fork opens up. Okay? But on this other lagging strand, every time you need a new fragment, you're going to need a new primer, right, to get the DNA replication started. Right?
When we start with the RNA premise, the area that we have complemented with an RNA, we do it again with the DNA? We should do it again. For instance, you said that around
10 RNA bases, yeah.
10 RNA bases. So there will be a set of 80 GCs that we are 10, approximately 10 of them that we will be complementing with an RNA.
Right.
But then when the DNA polymerase
Yeah. No, you have to fix that. You have to fix that. That is going to be cut out, then fixed, okay? Yeah, I'm getting to that. Yeah, you cannot say you replicated DNA by leaving RNA, those 10 nucleotides of RNA in them. No, you cannot, okay? So that has to be taken out. I'm getting to that. That's the next slide. So on this slide, right, we make the observation that an RNA primer on the leading strand, an RNA primer is needed only to start the replication at a replication origin. However, for the lagging strand, a new primer is needed every time a new DNA fragment has to be synthesized, right? And to produce a continuous new DNA strand from these many Okazaki fragments synthesized on the lagging strand, 3 additional enzymes are used. Right? First, you have to go ahead and cut out, right? You have to go ahead and remove that RNA primers. Then you have to go ahead and synthesize the complementary DNA, right? To fill in those 10 missing bases. And then you have to make sure that those things are linked together, right? You form the phosphodiester bond. So there are 3 enzymes that will be needed for doing that, right? And so the enzymes that are needed to produce continuous DNA strand from many separate pieces of RNA and DNA. First, you need a nuclease. Again, a nuclease is an enzyme which basically disintegrates nucleic acids, right? So nuclease which disintegrates the RNA primer. Then you need a repair polymerase, right? Which replaces the disintegrated RNA with DNA, right? And then you need something that is called a DNA ligase. To ligate means to tie things together. So you're now going to tie these separate strands together, right? These pieces together. So you need a DNA ligase which will join the 5' phosphate end of one DNA fragment to the 3' hydroxyl end of the next one, right? And that's how you will get a completely replicated DNA strand. All right. Any questions? Now let's do a listing of the proteins that are used in DNA replication. The most important one is DNA polymerase. Right? This is the protein that is at the core of the replication machinery. Right? In addition to that, you will need a primase, okay, to lay out the RNA primer to get the process started. Then you have a protein that is called helicase, which is a protein that uses the energy of ATP hydrolysis to move along the DNA, opening the double helix as it moves. Then you also have what is called a single strand binding protein, which will cling to the single stranded DNA that is exposed by the helicase and prevents it from immediately reforming base pairs. Because once you have separated the 2 strands out, okay, you want to go ahead and complete the DNA replication procedure, right? You don't want the thing to close back because it has a natural tendency to pair back. And then you have something that is called a sliding clamp, which will keep the DNA polymerase firmly positioned on the DNA template as the polymerase slides along, because you want to be making copies of the template, right? Or copies of the complement of the template. And on the lagging strand, for obvious reasons, the sliding clamp will have to release the polymerase from the DNA each time that an Okazaki fragment has been completed, right? Every time you completed it, then you have to get the DNA polymerase off the DNA template and then to a different location, right? And the detailed mechanisms by which that happens, that's not understood. At least it was not understood at the time when I first read the book. That's like, maybe 15 years ago. I don't know whether it's understood now or not, right? But if you take a look at the book, they have some schematic diagram about how all of this could be accomplished, like showing these different proteins and things like that. It's like a small machine that is, whose components are working together to accomplish the overall objective of DNA replication. Okay, any questions? Yeah, go ahead.
Opening the double helix as it moves, the helicase one.
Yeah, yeah.
This is at the beginning.
Yeah, at the beginning.
For separating the strands.
Yeah, because you need access to the template strand. Okay, you need to see what is in there before you can, synthesize the complement. Yes. All right, any other questions? Now, because all of the information specifying a living organism is contained in its DNA, cells go to great lengths to maintain the integrity of the DNA. And one aspect of maintaining this integrity is that DNA replication must be carried out very, very accurately, right? And the proofreading activity of DNA polymerase ensures, because it is checking its work, okay? Before putting in the next nucleotide, it checks the previous one. So the proofreading activity of DNA polymerase is going to ensure that in DNA replication, only about 1 error is made for every 10 to the power of 7. So that's like 10 million nucleotides that are copied, right? So the error rate is 10 to the power of minus 7, right? And elaborate DNA repair mechanisms will ensure that this accuracy is enhanced even further, so that only 1 error is made for every billion nucleotides that are copied, right? So the error rate is 10 to the power of minus 9. So roughly speaking, if you're copying the human genome, right, you'll make 3 errors, right? Huh? Yeah. It's a lot better. And in fact, I remember the first time I was reading the book on molecular biology, it gave a listing of how many errors the United States Postal Service makes, okay? How many errors the UPS makes, right, in parcel deliveries, and compare that with DNA replication. This is doing much, much better job, okay? Yeah. Yeah, lots of checks and balances. Yeah, lots of checks and balances are there. Now, despite these elaborate repair mechanisms that are in place, sometimes a permanent change in the DNA, which is called a mutation, can occur, right? Because there are three errors that are being made. Like maybe some of them might build up, they might propagate, right? So such changes over the long run, that is billions of years, have led to the wide variety of living species that we see on Earth today, right? However, and that confers basically survivability in a changing environment, right? Let's say the temperature of the world, went up by several degrees, almost all of us will be dead. Some organisms will survive and they will take over, right? That's what will happen. So, however, in the short run, if you have these DNA mutations, they can spell disaster for the organism, especially if it's a multicellular one, because your cells are going to behave in ways they should not be, right? For example, cancer is a disease that can result from the accumulation of DNA mutations. Okay, any questions so far? And I will talk a little bit about cancer, later on in this course. Now, before proceeding further, I want to introduce some terminology related to genetics and heredity, right? So a gene, okay, what is a gene? A gene is generally a stretch of DNA which codes for a protein, right? Then if you look at, papers like in genomics and all, you'll see the term allele. What is an allele? An allele is a variant of a gene, right? All of us have the hemoglobin gene, but we don't have the same allele because our DNA is different, right? An allele is a variant of a gene. So although different individuals may possess the same gene, the exact DNA sequences are not identical. That is, they possess different alleles of the same gene, right? A wild type allele is the kind that is normally present in the population, right? So most people have it. That's the wild type allele, okay? A mutant allele is one that differs from the wild type, okay? Again, I mean, the wild type allele, your DNA, my DNA is not the same, but if we both have the functional gene, right, we both belong to the wild type allele. That's the one that's generally present in the population. They have a lot of similarities, otherwise they wouldn't be having the same function, right? But the exact sequences are different. So a mutant allele is one that differs from the wild type. So in many situations, altering parts of the genome of an organism will not result in a change at the macroscopic or observational level, right? Such a change is called a genotypic change or characteristic. So if you make a change in the genome, that's a genotypic change, right? But among those 3 billion nucleotides, only about 2% of that codes for proteins, right? Rest of it doesn't do it. I mean, it's called junk DNA. We are beginning to discover its function slowly, right? But if you make some changes over there, there is no loss of function, right? So if you make a change in the DNA, right, that is a genotypic change, right? On the other hand, a characteristic that manifests itself at the observational level is called a phenotypic change or characteristic, right? So somebody's DNA change, that's a genotypic change. But after the DNA change, if that person gets cancer or a tumor, that's very, very much a phenotypic change because you can see that happening at the observational level. Or if the person falls sick, that's a phenotypic change. So genotype and phenotype, right? So few more terms. Now if you look at humans and many other organisms, each cell except the reproductive ones, they contain 2 copies of each gene. So except the egg and the sperm, all the cells, right, in a human being, they contain 2 copies of each gene because we are diploid organisms, right? Diploid means 2 copies of each gene we have, right? So consequently, in such organisms, if you go and mutate only 1 copy of a gene, that may not result in the manifestation of the associated phenotype, right? So let's say for the gene that codes for hemoglobin, right? If you go and mutate only 1 copy of that gene, right? You may not get sickle cell anemia because the other one is still producing some protein, right? Like I used to have a student here who was a carrier of sickle cell anemia, so he had 1 copy mutated. He told me he used to feel tired, but he's not like bedridden or something like that. Okay. So mutation where you need both copies of the gene to be mutated to see the change manifested in the phenotype, that is called a recessive mutation, right? On the other hand, a dominant mutation is one where mutating only 1 copy of a gene is sufficient for the phenotype to manifest itself, right? That's a dominant mutation. And you can think of it as like if you're looking at a car, okay? You have 2 brakes. You have the hand brake and the foot brake, right? I mean, so if both the brakes are messed up, then you're in trouble, okay? But you have only one accelerator, right, or the gas. So if that accelerator is messed up and it's accelerating the car, you're in trouble too, okay? So you could think of the brakes as something recessive, right? And the accelerator as something that is dominant, right? So a mutation over there, a single mutation in a dominant situation will cause problems, right? In a recessive situation, you need both the genes to be mutated, right? Before you see anything anything that is significantly observable. Now, when the two alleles for a particular gene in a genome are of the same type, that genome is said to be homozygous for that gene, right? So you have both wild type or both mutant, right? Then it's homozygous. On the other hand, when the two alleles are of different types, right? Then the genome is said to be heterozygous for that gene. So one could be wild type, one could be mutant, right? Now, there are many diseases that can be caused as a result of DNA mutations. For example, sickle cell anemia is an inherited disease that is caused by a single DNA-based mutation in the beta globin gene. Remember, I've been saying all along, right? In hemoglobin, there are two alpha subunits, there are two beta subunits, right? And for that beta subunit, that is coded for by the beta globin gene, right? And the beta globin gene codes for 146 amino acids. You mess up one DNA base, right, in the beta globin gene, you get the wrong amino acid. One out of 146 is wrong. Hemoglobin doesn't have the right shape, right? And you can get sickle cell anemia if both your copies are messed up, right? If only one copy is messed up, then you're a carrier of sickle cell anemia, right? You could feel tired. Your energy level might not be that high, right? But you will not have the full-blown disease. So humans have two copies of this gene. Like all the other genes, they have two copies. One inherited from each parent. So to have full-blown sickle cell anemia, one must have two defective copies of this gene. So if both your parents, are carriers of sickle cell anemia, and each one of them has given you a bad gene, then you are in trouble. You have full-blown sickle cell anemia, and that is catastrophic. Now, if an individual has got only one defective copy, then he or she does not show symptoms of the disease, although they can pass it on to their offspring with another person having a defective copy of the same gene. Right? So that's why, if there's inbreeding and all that, there's a problem, okay? Because, the chances of somebody getting two copies of that defective gene are pretty high. Right? So a person with one defective beta globin gene is a carrier of sickle cell anemia, and clearly the mutation that is involved in sickle cell anemia, it is a recessive mutation. Why? Because you need both the copies to be mutated for you to get the disease, right? That is sickle cell anemia, or to get the observable phenotype, right? So that's a recessive mutation, right? And the carriers of this disease, they'll have one wild type gene, one wild type normal hemoglobin gene, right? And the other one will be a sickle cell gene, right? So they have a genome which is heterozygous for the beta globin gene. Right? Again, this is just an example to illustrate the terminology that we just introduced. Another disease that can result from DNA mutations is colon cancer. So colon cancer is caused by the failure to repair DNA damage in the colonic cells. Right? Remember, the cells on your skin are replaced once every 10 days. The cells lining your gut, they are replaced every 3 days. Right? Now, if the bad guys stay on, then that can lead to cancer. Right? So you don't want cells with damaged DNA to propagate, right? Because that can cause problems, right? So colon cancer is caused by the failure to repair DNA damage in the colonic cells, because these are being turned over every 3 days, right? So the DNA better be repaired, right? You don't want the mistakes to propagate. Now humans inherit two copies of a DNA repair gene, one from each parent, right? So let's say one of your parents gave you a defective copy, right? So you got a defective copy from one parent. That means you just have one more to go, right? If somehow through DNA mutation the other copy gets mutated, you are in trouble big time, right? That's when you would get colon cancer. So for most people, they have two copies, so it's okay, and it will take a longer period of time for both of them to get mutated. So that's why, typically for colon cancer, people check for that at the age of 50 and above, right? You don't do regular, colon screenings on 20-year-olds, right? So the incidence of colon and other types of cancers goes up dramatically with age, since unfortunately the DNA repair mechanisms, they slow down as one ages, right? So mistakes will accumulate, and and there could be mistakes in a crucial area that could lead to cancer. And also enough mutations have to accumulate before cancer results, and so time is required for the initiation and progression of the disease. Because before cancer can happen, like a cell would have to acquire maybe 5 or 6 independent mutations, because there are lots of different checks and balances, right? Because a cancer cell is like a rogue cell, right? The other cells will try to kill it, right? But then it can become deaf to those signals and then take over, right? But the DNA mutations do play a component. Like even for breast cancer, there's a particular gene called BRCA1, BRCA2, right? If somebody has those genes mutated, the risk of getting breast cancer is much, much higher, right? Now, without DNA mismatch repair, mutations would develop much faster and the incidences of cancer would go up catastrophically. The DNA mismatch repair system recognizes the errors in the DNA replication and corrects them and thereby prevents them from propagating and possibly causing a cancer. Right? It is not yet known how the DNA repair machine preferentially acts on the newly synthesized strand and leaves the original strand unchanged. And again, not yet known means today it might be known. I'm talking about stuff from 15 years ago. Right? I don't have the most updated copy of the book. Right? So it might be known right now. Now, how does DNA get damaged, right? So one thing is there can be mistakes in replication. But in addition to replication errors, the DNA in our cells is continually suffering damage, either spontaneously or due to agents such as sunlight, chemicals, etc. And examples are what are called depurination and deamination and ultraviolet radiation in sunlight. And I will talk about that on the next few slides, right? What is depurination? Now remember, purines are adenine and guanine, right? Right? So you have purines and pyrimidines, right? So in depurination, which occurs spontaneously and in large numbers, a purine base, which is adenine or guanine, is removed, resulting in a DNA strand that has got missing bases, as shown in the figure on the right. So this was a guanine, right, that was attached to the sugar phosphate backbone. Spontaneously that guanine got removed, right? So this is like a situation where this strand it's like missing teeth, right? So now if this strand is used as a template, right, for synthesizing a new DNA strand, guess what? Anything could be put in this location because the base is missing, right? So the chances of mistake are high. Okay. So when such a strand with missing bases is used as a template, replication errors are bound to occur, right? So this is depurination. Another type of thing that happens is called deamination. That means removal of an amino group. An amino group is lost. Example, you have a cytosine here. This is the cytosine, the sugar, right, deoxyribose phosphate, so sugar phosphate backbone. All right, so this cytosine loses an amino group. NH2 is gone, all right? And then so when it loses that amino group, it now becomes uracil, right? You can check the structures, all right? When you take the amino group away from cytosine, it is uracil, all right? Now C pairs with G, right? Cytosine pairs with guanine, right? So it pairs with guanine, and uracil pairs with adenine. Okay, so if that strand is used as a template, you are going to get mistakes. Right? So you'll get the mistake that is referred to as base substitution. Right? Another way in which DNA mutation can occur is every time you step out into the sunlight. Right, the DNA in your skin cells is getting mutated. Why? Because the ultraviolet radiation in sunlight, it can damage DNA by causing the formation of covalent linkages between the adjacent thymine bases, thus forming what is called the thymine dimers. Like some linkages are established here, right? So these have to be corrected, because if they are not corrected, you will get skin cancer, right? Now the good news is that the information in DNA, okay, it is stored on 2 strands, okay? So hopefully the sunlight didn't mess up both your strands. If it messes up both of your strands, then that's bad, okay? Because then there is no template to go and see what the correct information is, right? So nearly all of the DNA repair mechanisms depend on the existence of 2 copies of the genetic information, one in each strand of the DNA double helix. So most DNA damage creates structures that are never encountered in an undamaged DNA strand. And this is thought to facilitate the identification of the damaged DNA strand. And the basic pathway for repairing DNA damage involves 3 steps. Again, you'll figure out the damage, cut out the damaged portion, right? Then use a repair polymerase to fix it, then use a ligase to stitch it back together. So the DNA damage is recognized and removed by one of a variety of different nucleases. Again, nucleases are enzymes that disintegrate nucleic acids, right? Then a repaired DNA polymerase binds to the 3' hydroxyl end of the cut DNA strand and fills in the gap by making a complementary copy of the information that is stored in the undamaged strand, right, or in the undamaged template. And when the repaired DNA polymerase has filled in the gap, right, a break remains in the sugar phosphate backbone, right. So this nick in the helix is sealed by DNA ligase. Okay. Again, as I said before, ligate means to tie. So one thing good about biology is, from the name of the enzyme, you will know its function, like oxidase, reductase, dehydrogenase, and so on. Okay. ATPase, hydrolase, all right. Phosphatase. The only one you don't know, at least I think is not obvious, is kinase. Okay. What does a kinase do? Anybody? Huh?
It speeds up the
No. Hey, you cannot tell from the name. What does the kinase do? Anybody? What does the phosphatase do? Yeah, so but what does phosphatase do? Huh? Which one removes the phosphate? Phosphatase removes the phosphate, kinase adds the phosphate. Oh, yeah. Anyway, keep studying. Okay, any questions? Now, failure to repair DNA damage can result in serious diseases. For example, failure to repair thymine dimers can result in the disease which is called xeroderma pigmentosum, right? This is a disease, like if kids have this disease, they can never play out in the sun, right? These kids cannot get out into the sunlight because they'll get blisters all over and skin cancer. So kids that have this disease, they have to go and play out at night, okay, when there is artificial electric light. They cannot they cannot lead a normal life, right? And people with this inherited disease are prone to severe sunburn and skin cancer, right? This is just one example, right? There are many, many other diseases that are caused by mutations in the DNA or the failure to repair damaged DNA. So if we did not have, these repair mechanisms in place, one trip out in the sun will probably get you skin cancer, right? Or you're in trouble, right? Sunburn and things like that. So fortunately, we have these repair mechanisms, but they slow down with age, right? So that's why they tell you to avoid the sun, right? Because, if you overdo it, you're just sowing the seeds for skin cancer, yeah.
I saw a really interesting paper about why that happens. Turns out that your genetics actually are like the genetics of the human race are geared towards producing more humans, and so the thing is they're optimized for when we're younger instead of when we're older. So there's all sorts of
Yeah.
property ways to get
Yeah. Yeah, basically, without all this modern medicine and all those interventions, probably you're supposed to live till the age of 40, okay? I suppose all of you are below 40, so you don't know, but typically people, once they cross 40, then it's like, your body becomes different, okay? You cannot do the same things and get away, okay? And like one of my colleagues used to say, 40 is the time for you to basically exit the stage, okay? The next generation takes over. But now with all the modern science and all that, the life expectancy, whatever, 70, 80 and all that, but you will have those problems, yeah. Because you're trying to do something different from what nature intended it to be, yes. That is correct. So in conclusion, I'd like to state that the high fidelity with which the integrity of the DNA is maintained means that closely related species have got very similar DNA sequences, right? For example, humans and chimpanzees, they have DNA sequences that are about 98% identical, right? Even apparently unrelated species such as humans and whales share a lot of similarity in their DNA sequences, maybe as much as 95%. I forget the exact figure, right? And this strengthens the hypothesis that they must have all originated from a common ancestor at some point in evolutionary time, okay? Because the DNA sequences are so similar. Okay, any questions? Yeah, we have some time, so I will move on to the next chapter. But I'll be happy to take any questions that you might have on this chapter. So the next chapter is about transcription and translation, right? So I said that the information in the DNA codes for proteins, right? So how do you go from DNA to protein, right? That's what this chapter is about. Okay, what is the code that allows you to go from DNA to protein, right? And how is the information from DNA, first copied into another molecule called RNA, and then translate it into protein. Right. So the DNA that is contained in the genome of an organism primarily codes instructions for making proteins. Right? And recall that there are only 4 DNA bases: adenine, guanine, cytosine, and thymine. Right? So an instruction that is written out in the language of DNA is basically an instruction that is written out in a language that has a 4-letter alphabet, A, G, C, T. Right? It's a message that is written out using only A, G, C, T. Right? On the other hand, proteins are made up of 20 different amino acids, right? 20 different amino acids that are linked together by peptide bonds. So since there are 20 possible amino acids that occur in nature, the amino acid sequence for a protein can be thought of as a biological message written out using letters from a 20-letter alphabet, right? And a natural question that comes up is, how does the DNA message coded in the 4-letter alphabet map into the amino acid sequence of the appropriate protein? So in this chapter, we are going to provide a detailed answer to that question. Right? So how do you go from DNA to protein? Now it turns out that the DNA does not direct protein synthesis by itself, but it acts through certain intermediaries. Right? So you do not go directly from DNA to protein. Right? You go from DNA to RNA and then to protein. So when a particular protein is needed by the cell, the nucleotide sequence of the appropriate portion of the DNA molecule is going to get copied into another type of nucleic acid, which is called RNA or ribonucleic acid, and we have already seen that. Okay. Now for RNA, all right. So what are the nucleotides that you can have in RNA? What are the bases that you can have in RNA? All right. And what is the sugar? Not you, somebody else. What is the sugar that you're going to have? Ribose, yeah. And then you have the phosphate. All right. So those are the units that make up RNA. RNA is not as stable as DNA. All right. RNA disintegrates very quickly because it's produced on an as needed basis. In fact, even on our skin and all, we have what is called RNAs, that is RNA disintegrating enzymes, right? So that when bacteria and viruses invade, right, we can actually, disintegrate their RNA, right, before they can make us sick. So those levels of protection are there. So RNA copies of short segments of the DNA are used as templates to direct the protein synthesis. So the portion of the DNA that needs to be, kind of translated into protein is going to first get copied into RNA, again using complementary base pairing, right, from the template, and this process is called transcription, right? And then from the RNA, when we make protein, that is called translation. So again, the process of copying the DNA into the appropriate RNA strands is referred to as transcription, and the process of producing the protein from the information in the RNA is referred to as translation. Right? So transcribe means to just copy in a different language, right? Slightly different language, right? Now some terminology, when a gene is being transcribed, right, that means the corresponding portion of the DNA is getting copied into RNA, right? It is said to be expressed or to be turned on. Okay. And the usual flow of genetic information is from DNA to RNA to protein, right? And this fundamental principle is referred to as the central dogma of molecular biology, right? All organisms express their genetic information in this way. I mean, there are some exceptions, like if you look at, HIV, right, that causes AIDS. That's an example of a retrovirus where there is RNA, it will go from RNA to DNA, so it has reverse transcription. We'll talk about that later. Right? But in general, pretty much all organisms, the information from the DNA is first copied into RNA, and then it is translated to protein. And this is referred to as the central dogma of molecular biology. Right? All cells, from the simplest bacterium to complex organisms such as humans, they express their genetic information in this way. So the use of an RNA intermediate makes it possible to more rapidly produce large amounts of a particular protein. Right? This is because many identical RNA copies can be made from the same gene. Right? Because if you have one master copy, right, then you can make many copies, right, and then copy from those. Right? That way you have an opportunity to make many more copies at the same time. Right? Instead of just copying from the master copy, which is going to kind of limit your speed of copying. Right? So each RNA copy can be used to simultaneously produce many identical protein molecules. So by controlling the efficiency of transcription and translation of the different genes, a cell can produce small amounts of some proteins and large amounts of others, because the cell doesn't need the same amount of each protein, right? Like for example, if it's a red blood cell, it needs hemoglobin, right? If it's a liver cell, it doesn't need hemoglobin, right? So it shouldn't be producing that, right? So the cell has mechanisms to control that. In addition, a cell can change gene expression. That means copying the corresponding stretch of DNA into RNA in response to the temporarily changing needs for a particular protein. So over time, as the needs for a particular protein change, right, the cell can, control the transcription of that gene, right? And a very good example of that, which we will encounter in the next chapter, is what is called the lactose operon, right? Where, like bacteria, if you put them in an environment that has glucose, right? They are producing they are producing proteins that are needed to metabolize glucose, right? They do not turn on the genes that are needed to metabolize lactose, right? However, if you remove glucose as the food source and you put in lactose, then they start producing the enzymes that are needed for breaking down lactose, right? And this was discovered in the 1960s, and again, this was the topic of another Nobel Prize, okay? Because this is how genes, how cells regulate gene expression, right? That's what it showed, right? Any questions? So DNA and RNA, they're similar and also they have differences, right? So RNA differs chemically from DNA in two aspects. The first one is that the nucleotides in RNA are ribonucleotides, right? That is, they contain the sugar, ribose, right? Instead of the sugar deoxyribose that is present in DNA. Okay, again, nothing new. If you have been reading the notes, you've listened to my lecture and all that, this is stuff you already know, right? And the other difference is that DNA will contain the four bases, adenine, guanine, cytosine, and thymine, while RNA contains the bases adenine, guanine, cytosine, and instead of thymine, it will contain uracil. Now, since uracil pairs with adenine, the complementary base pairing properties described in the last chapter for DNA apply also to RNA, right? So therefore, in RNA, the base pairing is going to be A is going to pair with U as compared to A pairing with T in DNA, and G is going to pair with C, which is the same as in DNA. Okay. However, so despite these similarities, DNA and RNA differ quite dramatically from each other in their overall structure. Right? DNA always occurs in nature as a double-stranded helix, right? And very, very stable molecule. Even the criminal tries to, get rid of their DNA and all that is difficult, right? Set fire, do whatever, so they invariably leave behind some DNA that can be used to track them. On the other hand, RNA is a single-stranded molecule and it will disintegrate right away. Unlike DNA, a single-stranded RNA chain can fold up into a variety of shapes by like hydrogen bonds between the different base pairs, right? Just as a polypeptide chain folds up to form the final shape of a protein. So an RNA can store information in its sequence of nucleotides. It can also serve as a catalyst because it could form, different three-dimensional shapes. So that's an important difference between DNA and RNA. So RNA can not only store information in the sequence of its nucleotides, like DNA, but it can also serve as a catalyst in certain situations or as a part of a larger molecular structure. And the following figure shows a single-stranded RNA molecule assuming a particular shape resulting for the formation of hydrogen bonds between complementary base pairs, like shown here. Okay, so you can see that U pairs with A, right? You can see that U pairing with A, G with C, and so on, right? So because the RNA molecule is single-stranded, so parts of it can form hydrogen bonds, right? And it can form some three-dimensional shape, and it might even function as a catalyst. In fact, people think that life originated in RNA world when RNA did everything, right? RNA, served as a catalyst, and it also served the function of information storage, right? So over evolutionary time, the role of catalyst was given to proteins, right? And then DNA became a much more permanent storage of information, right? And so it's like a division of labor, and RNA became the intermediate. So RNA was the main guy before, right? But over time now, RNA just became like the middleman. So that's what people think. So I don't know whether that's true or not. Okay, I think let's stop here for today. We have a few minutes, but we've made good progress, right? And I don't want to inundate you with information. I could have gone on, but Next time we will start talking about transcription and translation. Right? Are there any questions?